Very pleased today to have with us again Dr. Antonio Citro. We've been talking with him sort of since the beginning of this story. The talk today is bioengineering of a human iPSC-derived vascularized endocrine pancreas for type 1 diabetes, and he's coming to us from Milan, Italy. A little bio about him. He's the project leader and principal investigator of the Pancreas Bioengineering Lab at the DRI San Raffaele Hospital in Milan. And he was trained in organ decellularization and bioengineering in the OTT lab at the Harvard Stem Cell Institute in Boston. His current research builds on his expertise in beta cell replacement and regenerative medicine to bioengineer the endocrine pancreas microenvironment ex vivo. In this context, he has recently explored the combination of endocrine cells, for example, neonatal pig islets or iPSC-derived beta cells with bioengineering platforms. He serves as the chair of the International Pancreas and Islet Transplantation Association, otherwise known as EPITA, Young Investigator Committee, and is a member of the Young Investigator Committee of the International Xenotransplantation Association, or ICSA. I'm going to drop the link to his paper in the chat, and we'll take questions during the talk. You can drop them into the chat. We'll also have a little bit of time at the very end. So welcome, Antonio. Thank you so much for joining us. This is a really exciting avenue of work, and we're really interested for you to unpack your recent findings and current, research in the laboratory.
Thank you, Monica, for having me. This is my third time, so it's a repeated story. And good morning or good afternoon, everybody. It depends where you're located. So it's my pleasure to introduce you our recent paper that is like the last chapter of a long story, as Monica already mentioned, about bioengineering our decellularized scaffold for beta cell replacement. So, oops. Wait a second. Okay, so my first disclaimer is that we are using, as I mentioned, organ decellularization technology. And this is, for people that are not familiar, this technology allows you to strip cells out from the organs, preserving structure and architecture. So originally, this technology was invented in order to reduce the organ demand. So you can decel organs that are discarded from transplantation and then decelling them, repopulate it with the cells that you want, and then restore their function and make them eligible again for transplantation. We have a long story in terms of experience of organ decellularization. We worked in collaboration with Wake Forest in 2014, Giuseppe Orlando and Andrea Peloso, in which we demonstrated it's possible for the first time, actually, decellularized human pancreas. We have worked also with kidney, heart, and lung. This is just to say that if you are able to preserve vascular access of different organs, you can decel any organ in your body and then replace the original function, transplanting, seeding within the organ using a bioreactor, different cell types. And this is the technology that actually we are using for beta cell replacement strategy. So I don't want to make this story long, because I think that all the audience knows the need of the scaffold for beta cell replacement. I want just to highlight the fact that when we isolate the islet from their own specific microenvironment, we completely disrupt their connection with extracellular matrix and vascular network. And at the end of the process, what we have in our hands is an inflamed preparation. And when we transplant back this preparation to the portal vein of type 1 diabetes patient, we have a donor inflamed tissue, donor recipient inflamed place. And during this process, normally we request to the islet to engineer de novo their own specific microenvironment. And during this time, we're going to lose from 50 to 70% of the islet mass. So in Milan, in the last decades, we tried a lot to modulate in vivo the engraftment. So we try, for example, to modulate the recruitment of polymorphonuclear cells after transplantation into the portal site. This was successful in preclinical model of transplantation, but was not that successful in a clinical arena. Then we tried to modulate macrophages, blocking MCP1, for example, after transplantation in preclinical model. And then we tried also to mirror the immunosuppressive regimen after transplantation in preclinical setting. thanks to Lorenzo Piemonte that identified also a possible extrahepatic site, we also tried to transplant islet into the bone marrow and locally irradiate the bone marrow in order to improve the space for transplantation. But if you look all these efforts together, it's quite easy to understand that working in vivo in modulating engraftment after transplantation is a sort of an unfair competition, because although you can block a specific pathway, there is a subsequent pathway that can bypass your inhibition, and then you lose actually your power in terms of controlling engraftment. So it's quite easy to understand that we are missing a piece of the puzzle in terms of islet engraftment. And in order to understand from bioengineering perspective, which was this piece of puzzle, we look at the pancreas, in which we nicely, as you can see here by this fantastic review by Sharon Stabler, you can see that the islets are surrounded by a dense vascular network and extracellular matrix that we call endocrine niche. So if we want to let the islets engineer the endocrine niche in vivo, we're going to lose time, we're going to lose mass, and this is the reason why, from my perspective, we're going to lose a lot of engrafted cells over the first week after transplantation. So in beta cell replacement, there are a lot of bioengineering structure that have been generated recently in order to solve the problem of islet engraftment. I was invited by Breakthrough with a lot of fantastic people to write this review in which we try to summarize the recent effort in order to engineer scaffold for beta cell replacement. And we can easily summarize those in two main categories. The first is the self-assembly one, so easy one to make it, but less complex in terms of architecture. And then we have the functional vascularized architecture. These are much more complex in terms of architecture, but they are pre-vascularized in a sort of improving the engraftment ex vivo. So we start working in this direction. So we want to modulate the engraftment ex vivo prior to the implantation. And we are working, as I mentioned before, with decellularized scaffold. Here there is a spoiler because, of course, we are using the lung as recipient scaffold. So in 2019, as Monica mentioned before, we published the first paper in which we demonstrated that it's possible to biofabricate what we call vascularized islet organ for type 1 diabetes. The idea was to use a decellularized rat lung left lobe that have been repopulated with UVEC cells through pulmonary artery and vein, and rat islets by trachea, and the endothelial cells together, the pancreatic islets. Just to give you an idea how it looks like, for example, a lung from a pig after decellularization, this is how it looks like. You can preserve all the vascular access and use also a pig lung as a recipient scaffold for beta cell replacement. After 7 days of culture in our customized bioreactor, we were able to show that we are able to fully revascularize the scaffold with embedded pancreatic islets that are engrafted prior to the implantation. We tested them in vitro and we show an improvement in terms of insulin secretion. And this was also confirmed in vivo in preclinical model of NSG diabetic recipient mice, in which, as you can see by the blue line, the scaffold was able to revert immediately the hyperglycemic condition and they preserved the normoglycemic status till the entire follow-up. So we recognize that this technology has the potential to move forward, and we want to customize it for clinical translation. So we know that it's composed by 3 main compartments. So we have vascular compartment, extracellular matrix, and of course the endocrine compartment. All these 3 compartments can be separately engineered in order to achieve, to reduce this gap for clinical application. So in terms of autologous endothelial cells compartment, we can use autologous endothelial cells for engineering. So we can use what we call blood-growing endothelial cells, so endothelial cells that you can easily isolate from a single peripheral blood sample of subjects, or iPS differentiated endothelial cells. For the endocrine compartment, we can use neonatal pig islets or iPS differentiated beta cells as a limited source of beta cells. And then we also can work in modifying the extracellular matrix in order or to improve the cell function or to protect the scaffold against immunological rejection. So in 2023, we published the previous paper in which we demonstrated the combination of both cells and neonatal pig islets works with this technology. But today I'm going to present you the data about the combination of iPSCs, differentiated endothelial cells, and beta cells. So as a first disclaimer, I want to say that we use commercial iPS differentiated beta endothelial cells because the idea was not to test the power of the cells, but the feasibility of the technology using standardized cells in our system. So the first point was, can we recapitulate sort of pancreatic islets? So the idea was to combine these iPS differentiated beta endothelial cells in a ratio of 90 to 10%. And then we cultured them in these spherical plates with a conic shape in which you can oblige the cells to aggregate. So after a few days in culture, the cells start to aggregate and transform themselves into spheroids. We call them VIBEs, so vascularized A-beta spheroids. We track them during the engineering process. So in red, you have the endothelial cells, the iPSCs from endothelial cells. This is an input site live tracing. And you can clearly understand that after a few days, actually a few hours during the culture, we have the spheroids reaggregated and they generate what we call vascularized A-beta spheroids. We track the persistency of the endothelial cells over the culture time, confirming that those endothelial cells are still present till the entire culture. And of course, we characterize them in terms of chromogranin, C-peptide, NK6.1, and PDX1. And we confirm the presence of all of these cells within our spheroids. So they are functional vascularized beta spheroids. So the idea was to combine those spheroids with our protocol that we have already published. So the idea was to seed 30 million of iPS differentiated endothelial cells through pulmonary artery and vein of the lung, day 0, and then at day 1, combine iPS differentiated endothelial cells, 10 million, together with 2,000 of vascularized beta spheroids through the trachea. After 7 days of culture in our customized bioreactor, we want to evaluate, first of all, compartment generation in the scaffold. So the first was to assess the vascular compartment structure and function, and subsequently, the endocrine compartment, preservation and function. So let's talk about the vascular compartment first. So we want to establish if our culture media protocol is able to generate the vascular compartment of the scaffold again using iPSCs, endothelial cells. This was not confirmed yet in literature. So the first point was to test fluorescence microangiography assay. So this is a test in which you can perfuse through pulmonary artery blue beads immersed in an agarose solution, and then you can cast the lung and then evaluate if the beads can leak through the vascular tree in the airspace, or if the beads can be trapped in the vascular lumen, suggesting that you have a functional tight endothelium. So as you can see here, this is in CD31, the iPS endothelial structure. The beads were completely trapped in the vascular lumen, suggesting that we have a constrained tight endothelium that is able to perfuse in a 3D way the culture media through the pancreatic islet. And then we also challenge again the scaffold with dextran perfusion assay. Here, if the endothelium is tight, you have a faster dextran elution. And as you can see, this is confirmed by the same scaffold generated with iPS differentiated endothelial cells. So we confirm that we have a functional vascular compartment with tight endothelium, also in the presence of iPS differentiated endothelial cells. The second point was to evaluate the endocrine compartment and function. So thanks to the fact that we have an organ in a bottle, the advantage of the technology using a bioreactor is that you can perfuse through pulmonary artery low and high glucose, and then collect by the outflow, that is the vein, the insulin that is accordingly produced and measured by the ELISA. So in this case, we challenge the spheroids in a standard culture condition versus the organ culture for 7 days with the same batch match spheroids. So as you can see here, we do not only have an improvement in terms of overall insulin release during this test, but also the acquisition of a dynamic biphasic insulin secretion kinetics. After that, we also confirmed the engraftment of the spheroids within the vascular network. So as you can see here, we have those spheroids that are completely embedded within the engineered de novo vascular network within the IVAP. So we can conclude that also the vascular compartment was successfully engineered, and we can improve actually the endocrine function ex vivo prior to the implantation, suggesting an important take-home message. So the vascularized ECM is a really potent inductor of endocrine functional maturation, because as we know, the iPS-differentiated beta cells are not perfectly mature, they need time to achieve functional maturation, especially in vivo. But here we demonstrated already in vitro we can improve their function. But say that you have a functional organ ex vivo does not mean that you have functional organ in vivo. And that's why we move in a preclinical model of diabetes. We use NSG, diabetic recipient mice. And the idea here was to compare a pre-vascularized site, so the famous deviceless implantation site that was published some years ago in Nature Biotechnology by Andrew Pepper and Shapiro, versus a pre-vascularized scaffold. The IVEP was split into pieces and implanted under the unmodified subcutaneous space. After 14 weeks, we explanted tissue and evaluate the function of the scaffold after implantation. Of course, these are immunocompromised NSG mice in this case, and future experiments are ongoing to evaluate also in immunocompetent mice. So if you look at the glycemia curve here, as you can see by the blue line, the scaffold was able not only to achieve immediate function after a few weeks of implantation, but as you can see, preserve the normal glycemic status of the mice till the entire follow-up. We measured the C-peptide level in the first week after implantation in order to confirm that the exceptional function that we achieved in vitro was also achieved in vivo. And as you can see, the C-peptide increased immediately upon implantation. And when we explant the graft after 4 weeks or after 13 weeks, we immediately observe the reversal of the hyperglycemic condition, suggesting that the normal glycemic status was really driven by the implantation of the scaffold. And when we explant the graft, as you can see here, we were able to evaluate the positivity to chromogranin, the presence of the vascular structure that was human, so we preserved the iPS differentiated endothelial cells within the scaffold, and of course the insulin positivity within the scaffold. So which are the take-home message that we can obtain by this manuscript, basically, is that the human iPS-derived vascularized endocrine pancreas show that the revascularized ECM, and I want to stress this point, is an important inductor of functional endocrine maturation, because we achieved an immediate function in vitro in vivo, suggesting that the vascularized ECM can really we are looking the mechanicistic behind this, but they can really boost the function of iPS differentiated beta cells. And it showed for the first time a superior ex vivo endocrine maturation and function compared to standard iPS differentiated beta cells, and a superior immediate function in vivo compared to several control groups. Finally, I want to, of course, thanks to the pancreas bioengineering team, especially Francesco Campo, Alessandro Neroni, Cataldo Pignatelli, Matteo Monieri, Alicia Chirichelli, Camilla Teruzzi, all the internal and external collaborators, founding agency, and all of you for your attention. I'm here for questions.
Thank you, Antonio. That was very short, sweet, and full of information. I wanted to open it up to people who are in the room. If you'd like to ask a question, please unmute yourself. I will start with some questions. Okay, so you've got this great, model. What about transplantation site in humans? So, because your model so far has been a mouse, and so what are you thinking about? There's been some new work coming from China regarding insertion of cells, the iPSCs, beneath the rectus fascia. Sort of historically, the omentum has been a great site for implantation because of its high vascularization. And then you mentioned maybe a sub-Q site. What are you thinking for this model?
Yeah, thank you.
Get towards, humanized models or non-human primates.
Yeah. So thank you, Monica, for the question. So the scaffold now that we have used is a rat, and we are now moving for different type of scaffold from large animals. And the idea now is to have, the first problem is to have a size that is matching the dose of the islets or the beta cells that you need for restore function in patient. The positive thing is that if you consider mammalian lung, for example, we have more than 30 million of alveoli available as a surface. And we do not need so much space, actually. We need only 700,000 of equivalent islets to restore function in a patient with stem cell more, that is for sure, but not 30 million. So ideally, with a mammalian lung, we can actually have enough space, well organized in terms of architecture. But in terms of site, if we are scaling up in a different model, I do think that Omentum, as you mentioned, or subcute, are still site available for transplantation for this kind of approach, especially because we are pre-vascularizing the scaffold. So I consider this as an advantage, although in terms of ATMP is challenging because you are putting within the scaffold two different cell types. So increasing the number of the cells that you're using for ATMP generation is normally a nightmare. But as long as these cells can be produced, for example, from the same subject, can be genetically modified, for example, you can bypass this issue. And in this case, I would consider that the combination of pre-vascularization and maybe the implantation in Omentum, considering the size of the scaffold, can be okay for transplantation in the future clinical application. Of course, we have to pass through immunocompetent preclinical model and also large animal model to prove the efficacy. But we are confident since the data that we observed independently by the endocrine source or endothelial cells that we are using, because we combine neonatal pig islets in the first paper and now iPSCs, differentiated beta cells, we observe the same performance. And this is a very good backbone to scale up the system for a future clinical perspective.
Yeah, that all makes sense. Can Shaheen Rafi unmute yourself and ask your question?
Yes, thank you. In the last model that you showed, that in the lungs or or it was sub, it wasn't clear. The mice was cured, was subcutaneous. Yeah, where was this implanted?
Subcutaneously. So the lung was pre-culture and then split into pieces, okay?
Ah,
And then we make a task in a subcute space, a tunnel, surgically speaking was a tunnel in which we implanted the part of the inner part of the lung exposed to the subcute revascularized area. And then we put two different pockets, the two pieces of the lung. Because the idea was to transplant in the same site, so subcutaneously, the spheroids from iPSCs in a deviceless, so that is still subcutaneous but pre-vascularized versus the pre-vascularized scaffold. But the site is still subcutaneous space.
so the whole advantage of lung is the distribution of your islets, right? It basically segregates your islets into alveoli. That's the concept, right?
Yeah, and then we pre-vascularize the lung using both axes and also the trachea. So we cover entirely the structure of the D cell lung with the new vascular structure that was in this case iPS differentiated endothelial cells. So this was a protocol published, I think, in 2016 in Nature Biotech from Harald group, in which they demonstrate that it's possible to fully cover a D cell lung with endothelial cells using pulmonary artery and vein and also trachea in order to have a full coverage of the vascular network. And we use this combination of seeding islets through alveoli and then endothelial cells by this axis to have a revascularized islet niche, actually. So the alveoli are revascularized.
So why not just get your vascularized islet without lung, inject sub-Q? Why you have to go through this scaffold with the
So
lung?
what we did this experiment, actually, there was supplementary. It was not, I mean, the idea was not to put in this presentation, but actually what we demonstrated are two things. So we generate the same scaffold in the absence of endothelial cells, for example. So islet within the alveoli, no iPS differentiated endothelial cells, and we saw reduced function. And when we put the islet, the iPS, those iPS with endothelial cells implanted under the skin, we do not achieve the same C-peptide level upon transplantation. So we do not know actually which is the mechanistic effect of the vascularized ECM, but for sure, we observed this with neonatal pig islet and iPS differentiated beta cells. The vascularized ECM is an inductor of endocrine functional maturation. So we observed an improvement ex vivo in terms of insulin release and in vivo in terms of C-peptide release, glycemia correction. And the other thing is the more conceptual thing. So the way in which the lung is composed, from architecture speaking, is incredible advantage in terms of structure. So you can seed cells in a way that is already generated by Mother Nature, in which you have all the alveoli surrounded by the vascular network. You can still preserve in the bioreactor structure oxygenation of the cells with vascular oxygenation tubing, perfusion, dynamic culture. And so you can control the engraftment ex vivo in a not inflamed situation. We measure cytokines, for example. And this is another advantage in terms of using the lung ECM as template for delivering the endocrine cells.
I just want to mention, you showed that the dextran and all of those, but you know, islet endothelial cells are highly fenestrated. So I would argue here that the ECM in the lung is generating a non-fenestrated continuous lung endothelium, which doesn't phenocopy islet endothelium that are highly fenestrated without diaphragm. So I understand where you're coming from, but ECM of the endothelial cell respond to ECM. So you're generating a long ECM that may not mimic, although you show good C-peptide production. But question is, what happens over time with these endothelial cells? Because they want to behave like lung endothelium based on the ECM and not islet endothelium that is completely different. They transport fat, they're fenestrated. So the question is, did you look at those sub-Q, whether they acquire the features of endothelial cells?
Yeah, this is a perfectly a good question. So we look at long term, if we have still preserved human endothelial cells within the structure, and we observe an exchange with murine endothelial cells. So we observe murine CD34 endothelial cells that start to make a connection with the scaffold. And over the time, if we extend long term, I mean, more than 3 months, okay, the overview of the scaffold, we observe an exchange from human to murine endothelial structure. We do not observe the absence of endothelial structure, but we observe a progressive transition from human to murine in that case. But this is invasion from the human, from the endothelium from the recipient that looks fine to me, as long as we have like function. I mean, I understand, of course, your point that having I completely agree, actually, having like endothelial cells within a lung ECM, and maybe we are changing the phenotype of these endothelial cells. But as long as we can drive the function in a way that is supporting the beta cell function in vivo, this could be something which we can investigate, but still we can preserve engraftment in the first phase. That is the mandatory time in which most of the scaffold show and reduce power in terms of engraftment. Of course, this is my point of view.
Okay, thanks.
No problem.
Thank you. Can Marcus Muehlman, would you like to unmute yourself and ask your question? It has a few parts to it.
Absolutely. Hey, Antonio. Nice talk as always. Always a pleasure listening to your presentations. This is maybe not a new question, but like I'm always wondering, like when you start with the decellularized lung tissue, why the lung and why not pancreas? Because we all know like that the ECM niche, or especially the endocrine ECM niche, as you mentioned before, the specific composition of collagens, of laminins, is so well preserved and unique. What is the main reason why not to go with the other tissues like pancreas or something?
Thank you, Marcus. It's a pleasure also answer this question. This was at that time, I mean, 2019, actually, a strategic point, because the idea was not to use like a mesh digested ECM, but an architecture, I mean, the organ itself as structure for transfer the beta cells. As you know, pancreas is composed by less than 5% of the endocrine mass, and 95% is the exocrine part. So from bioengineering perspective, we do not need the 95% of that ECM. And the other part is from surgical perspective, the pancreas is really mushy, and you have a lot of connection in terms of microvasculature that you need to suture every time. So the probability to have a leaking tissue during the culture time is higher. In this case, we make a comparison in terms of ECM composition of the alveoli and the ECM composition of the endocrine ECM, actually. And we identified that all the protein that are present in the endocrine ECM, actually in the pancreas, are still present in the ECM of the lung. So we control collagen 1, collagen 5, and luckily there was in the alveoli, so the place in which we want to put the islets itself. So in terms of structure and also ECM composition looks perfect. And from architecture speaking, the advantage of the lung is that, as I mentioned, you have 30 million of the alveoli. So in terms of space, you can pull together like 1, 2, 3 pancreas together and deliver a huge mass. We do not think that we need this large amount of islets since we can improve engraftment, but this is the main reason from surgical and scientific point of view of selecting lung instead of pancreas. It's different, completely different story if you want to use like the pancreatic ECM as a sort of, let's say, with a disrupted architecture. You do not need like the structure itself. You want to use like, let's say, hydrogels or different things, you can use pancreas still. But in that case, 95% of DCM is not endocrine ECM. So I can still argue that which is the relevant part of this ECM for the endocrine function itself. So but this is mainly, these are the answer for our perspective in terms of strategic point of view.
Perfect, thank you. Maybe if I just kind of quickly go up with a follow-up question on that. It's maybe provocative in a way. I know IPS is the future, or are you going the right path? But I'm just asking, like, xenotransplantation is still like a very interesting approach. And since you're using animal-derived decellularized scaffolds, I mean, pig islets, for example, are quite easy accessible or quite in an unlimited manner right now. And do you think it could be in the meantime until the IPS therapies or approaches are advanced enough to be really used in a daily manner, let's say? Do you think like also xenotransplantation could work together with your approach, like using the animal-derived scaffolds, or what is your position or view on that?
Oh yeah, so what I can tell you is that we use both cell types. So we published the previous paper that we published, we were using neonatal pig islets. So we demonstrate that no matter if you are using neonatal pig islets or iPSCs, beta cells, the technology is working. So we have a plan A, plan B actually. But the idea is that I'm pretty in favor of using also xeno, especially because there are a safety concern. I mean, in general, using iPSCs. So the point is, iPSCs will be the future because in terms of, for sure, scalability, production, there are a lot of investors that are pushing in that direction, but also in the xeno world as well. In terms of genome editing, the pig can be edited from blastocyst level. So they are safer in terms of isolation, production, and so on. So I will be absolutely also in favor to test the technology in vivo using gene-edited pig islets, because this can be also a potential strategy, independently by the fact that iPSCs can work or not can work, or neonatal pig islets can work or not. I mean, this is something that we have worked on and we are working on. So both cells can work with the technology. So we are open to test both. And we know that they can be combined with endothelial cells. They can work in vivo. actually, when I stress the concept of the fact that the vascularized ECM can be an inductor of endocrine functional maturation, that concept came actually from the pig islets, from that paper, because the neonatal pig islets are not fully committed in terms of insulin secretion upon isolation. So when we combine with the technology, we observe exactly the same picture using iPSCs. So we saw in vitro immediately maturation, immediately insulin secretion. And this is a common factor that, from my perspective, put in the same cage iPSCs and xeno islets in terms of power for using this technology.
Thank you so much.
Thank
Fantastic.
you.
Fatma Dogan, would you like to unmute yourself and ask your question, or I can do it, whatever you prefer?
Thank you. Thank you for the great talk. I'm just curious a little bit more about clinical perspective. Using this decellularized membranes, what are the main clinical concerns and kind of reproducibility of these membranes for each time? And another question, actually a follow-up question with that, this decellularization steps are quite harsh. Actually, what are the main ECM proteins survive and remain functional after these procedures?
Thank you, Fatma. So this is a good point. So what I can tell you is that the decellularized porcine valves, for example, are actually used in a clinical application. And there are few companies that have requested FDA approval for using liver or like a lung from decellularized pig for clinical application. The major concern is the acute rejection that maybe the pig decellularized tissue can generate. But luckily now, thanks to the genome editing technology, the alpha-gal knockout pigs can be produced. So the ECM coming from alpha-gal knockout pig can use in this purpose. So you can reduce dramatically the rejection against the pig ECM. On the other side, fibrosis can be another concern. So something that you need to test in large animal is fibrosis. But there are a lot of technology that now allows you to modulate the ECM from in a chemical perspective to control the fibrosis. So you can engineer the scaffold in a way that you can protect the scaffold, not only by fibrosis, but in general from other type of reaction. The decellularization of answer to the point, which are the main protein preserved, is really challenging because it's really connected to the way in which you made the decellularization process. There are a lot of decellularization process. They are using detergent, without detergents. So it's really linked to the protocol that you are using. What I can tell you is that our protocol is able to preserve mostly of the protein within the ECM. And in terms of function, you can even test those. You can repopulate and see specific function, if they are connected or not with the ECM. We also made, like, for example, spatial transcriptomics in our scaffold repopulated in order to see which genome have been upregulated or not, in order to see if, for example, endothelial cells start to proliferate and then come activated based on the contact with the ECM. And I can tell you that this happen normally, but it's really dependent on the aggressive, if your protocol is really aggressive in terms of decellularization or not. For example, it's very complex and different if you're using detergent vascular perfusion in terms of decellularization or chopped tissue decellularized. Because in that case, you burn a lot of protein because you are much more aggressive in order to go to the core of the tissue in order to be decellularized.
Thank you. That's really interesting study. Thanks.
You're welcome.
Thanks. I have Kevin Chen. Kevin, would you like to unmute yourself?
Yeah, sure. Great talk. I was just curious, clinically speaking, how big is a scaffold?
So I missed the point. How big is the scaffold was the question? I assume yes.
Oops, sorry.
So
His question in the chat, it was, clinically speaking, how big will the scaffold be when transplanted into human patients? As the lung scaffold is fairly large, and where would you transplant it, which we kind of talked.
Yeah, so the rat one is not that big. You have to think that is 3 centimeter per 2 centimeter. If you move in pig scaffold, that would be, I think, the ideal shape for transplantation, is going to be 3 times bigger. Although that, you do not have to consider that we're going to use the entire lung. We're going to use only a single lobe or a specific unit that is smaller than the entire structure, close to the rat one. And in terms of space, it's able to harbor enough islets that you need to restore function. So ideally, I would say that the overall dimension of something that can go in clinical application is going to be from 3 to 4 centimeter per 2 centimeter. And these will be not, I would say, not bigger than the actual scaffold that have been already tested in large animal models, for example. But in terms of number, so numerosity, that is, I think, the most important part of the story, based on the fact that the lung has a lot of alveoli, as I mentioned before. We do not need a lot of scaffold to implant, actually, because in one single lobe you have 30 million of alveoli. So in that case, you can save. You have a lot of space to harbor enough islets to transplant. This is in theory. Yeah, we need to test, by the way.
Yeah, well, the surface area in the lung and it really is affords you so much space. They're just the way it's laid. So it's really ideal in that way. I did have a quick question for you. Have you considered, so I mean, you've got your beta cells and your endothelial cells combined in the transplant, but have you thought about doing an alpha cell, beta cell combinatorial organoid, or even an islet organoid that would be stem cell derived, not a xeno?
Yeah, I mean, this is something possible. We never did that. So ideally, with the concept to make spirits aggregation, you can aggregate all the cell type that you want. The bottleneck that I can emphasize is the media. So when you combine multiple cell types, it's really a matter of identifying the best media combination and the best ratio of different cell types in order to have a functional organoids or spheroids. This is, I think, a limiting factor. We travel a lot to identify which is the best ratio to get an improvement in terms of function, and which was the best media combination to preserve the beta cell function, but also not reduce the endothelial function as well. So it was really challenging. So ideally, from a conceptual point of view, it's possible. I'm not super fun to put a lot of cells within the spirits, really from technical point of view, because you need to address a lot of issues, but it's something possible and would be nice to have also alpha cells within the spirits. It's also important to say that as of now, iPS differentiated beta cells are polyhormonal cells. So you still have also glucagon positive cells within the preparation sometimes. So maybe it's, I mean, unless that you have like pure 100% insulin positive cells in your preparation, you need to add also glucagon, so alpha cells within the spirits. But at this moment, as of now, I don't see really the need based on the mixed population that you have sometimes batch to batch in terms of batch to batch variability.
Yeah, or you might even consider just having different alveoli populated with some alpha cells, some beta cells, different clusters. It's really an interesting setup you have here, and it's really exciting. I guess the last question I have for you is, in terms to scale to get to non-human primates and then to the clinic, what's that look like for you? And are you partnering with industry to make that happen?
Oh, yeah. We are not partnering with the industry. We are still in a research lab-driven institute, but we actually are working a lot in a computational analysis, because what we learned so far is that if you want to scale up the system, you need to recognize that if you are moving from a rat scaffold to large animal scaffold, no matter if it's like primate or pig, you are scaling up not only the size of the scaffold, but also the number of cells that you need to reconstitute, for example, the vascular tree, okay? So in order not to make like a very huge demanding experiment. We are really making computational analysis experiment in order to predict the number of endothelial cells, for example, or beta cells that we need in order to faster scaling up the system. And this is, I think, the future in terms of revenue, in order to save a lot of time, experimental setup. And I'm not saying that it's perfect, because now we are in the era of computational analysis, but it's something that can potentially help you to predict not the function, but the scaling up in terms of architecture of a complex device like this. And we are moving in this direction. And hopefully in the future, we're going to transplant the first scaffold in large animal models.
Okay, fantastic. Well, you've got some really great collaborators with Kevin Ott, Alice Tomei, Cherise Stabler. I mean, this is a really sort of a dream team to address this question and push it forward. So, and also congratulations, I'd say in the, when we first started talking, it was 2020. So in 5 years, your group has really sprinted and made a lot of discovery and moved this paradigm forward. So congratulations there. And we look forward to talking to you again, Antonio. This is always a pleasure. And thanks again for all the work you're doing in this space.
Thank you. Thank you for everybody for the nice interaction and conversation. Thank you.
Have a great rest of your evening. Bye-bye.
Yeah. Bye.